We now move on to the award of the JMT Medal and the lecture by the JMT medalist. The JMT Award is in honour of Sir John Myrick Thomas. It was named after Sir John Myrick Thomas, who was almost universally known as JMT. Before I present the award, I think probably most of you will be aware that Sir John sadly died a few weeks ago after a long struggle with cancer. There have been many very eloquent obituaries and tributes paid to Professor Thomas. I just really want to say three things briefly. The first is that John really was a giant in our field who made huge contributions. In fact, the current field of catalysis would be very different without the many contributions that John made. Secondly, that he was a great supporter of the Catalysis Hub and of the people who worked in the Catalysis Hub. Thirdly, that I think the whole catalytic community owes Sir John an immense debt of gratitude. And what I would like to do is now just to pause for a few moments in silence to honour his memory and his contributions. Thank you very much. And you see, you saw there briefly, Sir John presenting the JMT medal to one of the earlier awardees. Good, so the JMT medal is awarded annually by the Hub community, and this year it is awarded to Adrian Mulholland. Adrian has pioneered computational modelling of enzyme catalyzed reactions to reveal catalytic mechanisms and to analyze the effects of protein dynamics. His work has shown how simulations complement experiment in applications ranging from antibiotic resistance to enzyme evolution and design. Adrian is a very worthy winner of the 2020 John Myrick Thomas Medal. And I think Josie is showing the medal. There you can see it, and when we can next meet in person, we will present the medal in person to Adrian. So many congratulations, Adrian, and I would now like to invite you to present the 2020 John Myrick Thomas Award Lecture.
Thank you very much, Richard, and thank you very much to the committee. It is indeed an honour to receive this award. I first met JMT when I was a graduate student, and to my embarrassment, sitting next to him at a college dinner, we were chatting away, and I had no idea who he was. And I think it says something about him that he was not for one moment put out by the fact that this PhD student was unaware of the great man, and we just talked about chemistry, and afterwards I realized who it was, and he was enzymes. I was working on enzymes, or not his field, but his comments, his questions, and his suggestions were very useful. So it is really an honor to receive an award in his name. I hope about to start sharing my screen. If there is a problem, can you see that?
Yes.
Yeah, fine.
Good.
You haven't got it on the full screen. Oh, that's fine, Adrian.
So, no, I don't want subtitles, do I? I managed to switch the subtitles on. let me try again. Okay, so yeah, I'm going to talk about my work on modeling enzyme catalysis. So it was said a long time ago that to be an enzymologist, all you needed was some liver and a razor blade to chop it up. Now, believe it or not, I do have a razor blade, and I think I still have a liver. But what's changed recently is that it's now recognized that you really need a computer and you need to model mechanisms of enzyme catalysis to understand them. That's perhaps taken slightly longer in biochemistry than in chemistry, but I think it's now widely accepted. So just in terms of how did I get into the field? Well, working as an undergraduate with Gabriel Balint-Curti and Richard Dixon, as an incompetent in the lab, I was really struck by the idea that one could use a computer to calculate chemistry, that we could understand reactivity and predict experimental outcomes, product distributions, selectivity in the computer. We worked on F plus H2 reactive scattering, which is all very worthy, but I knew from having studied some biochemistry that I wanted to get into biological applications. I was lucky enough to work briefly at AstraZeneca. Well, it wasn't AstraZeneca, it was ICI Pharmaceuticals at Alderley Park, a great site where tamoxifen had been discovered and was under development. And it really showed me how basic chemistry could make a difference to people's lives, to drug development. And it also introduced me to understanding details of enzyme inhibition. So this enzyme over here, HPPD, we looked at, and we were able to identify it as a target for a herbicide and suggest other protein targets as well, showing that the herbicide worked by inhibiting the chemistry at the active site. Something else that was a recurring theme was that I recently went back up to Alderley Park. There are a series of plaques for the medicines that were developed by ICI and Zeneca. One of them being chlorhexidine, familiar in antibacterial solutions. And more recently, meropenem, one of our first-line antibiotics nowadays, but really bringing home, so the approval of meropenem came from 1994. Not many antibiotics have come to the market since then. And that problem of a lack of antibiotics and bacterial resistance to antibiotics is something I'll come back to later. So I wanted to combine my interests in reactivity modeling and biochemistry. And Rod Morrod at ICI Central Toxicology Lab put me in touch with Graham Richards. And Graham, shown in his pomp up here, recognizing my interest in reactivity and enzymes, suggested that it would be interesting to look at modeling reactions within enzymes based on the idea of Linus Pauling, that if you can design a compound that resembles a transition state analog, then it should compete and outcompete the substrate, the reactant, for binding to the enzyme active site. And potentially that would be a route to designing enzyme inhibitors. And many enzyme inhibitors are drugs or herbicides or fungicides. Alternatively, of course, understanding how the transition state is stabilized is a route to catalyst design. Potentially, we can use the principles that enzymes use to stabilize their transition states in non-biological or de novo protein catalysts. So yes, this was a good idea, and Graham gave me this paper by a guy called Martin Karplus and Ron Elber. Said this is a way to model transition states in large systems, and do this for an enzyme called triose-phosphatidylisomerase. There's a guy working in Oxford called Jeremy Knowles on an enzyme called triose-phosphatidylisomerase. Show this works, and this is a route to drug design. And lo and behold, if this works, we'll both do well out of it. And this all started very well, but within the first few months of my PhD, and I'd say this is a warning to graduate students everywhere, I found that my PhD had been done, that Martin Karplus had discovered the reaction pathway for triose isomerase using a method called QMMM, combination of quantum mechanical modeling with molecular mechanical modeling. This is one of the early applications of that technique to an enzyme. And having scratched my head for a while and talked to Graham, we thought it would be a good idea if I went and worked with Martin, and he was kind enough to host me. And later on, I went to work with him as a postdoc, and we did some nice work together on QMMM methods, which at the time were somewhat in the wilderness of computational chemistry, but of course became respectable in 2013 when Karplus and Varshall and Levitt received the Nobel Prize for QMMM methods, or in the broad sense, multi-scale methods, methods that combine loosely or tightly different levels of description of a chemical system. So we applied these methods to a few different systems, citrate synthase, charismate mutase, I'll come back to charismate mutase, DNA, and PHBH. Just picking out one of those because it was important, this enzyme PHBH, long name here, para-hydroxybenzoate hydroxylase. This is a flav-independent monooxygenase. And what was important here was that even with low-level QMMM methods at the AM1 level, we were able to show that the barriers we calculated for a series of substrates correlated with experimental rate constants. So the log of the rate constant correlated with the activation energy. So this showed that we had a predictive calculation, even at this low level of QM theory. So it was a predictive technique, and this was a success of QMMM. In absolute terms, these barriers are too high because of the limitations of the AM1 QM method that we're using. But it shows that the method and the transition state theory approach that underlies the prediction of the rate constant are realistic and applicable to enzymes. There's some debate about that. So these two enzymes, let me return to charismate mutase and PHBH. Long names, CM and PHBH for short, but fairly simple reactions. So a Claisen rearrangement reaction and an electrophilic aromatic substitution reaction for PHBH, but quite different chemistry. And working with Walter Thiel, one thing that we wanted to do was to see how accurate it is now possible to do calculations, QMMM calculations of activation energies in enzymes. And what these calculations showed from a few years ago with the late and much missed Walter was that it is possible to calculate activation energies for enzymes in a QMMM calculation at a level of accuracy that is equivalent to experiment, essentially. We can calculate reaction barriers at high levels of QM theory that are within chemical accuracy in agreement with experiments. In other words, they agree within 1 kilocalorie per mole with experiment for charismate mutase and for PHBH. That's only possible at the highest level of QM theory, in this case, local coupled cluster treatment. But we know from first principles that these calculations are accurate. These are a weak degree of approximation to the Schrödinger equation. Lower levels of QM theory give barriers that are in less good agreement with experiment. And I should say that this wasn't just activation enthalpies, but also free energies, which we can calculate in a composite way using QMMM molecular dynamics simulations at a low level of theory. The important point here is that now in a QMMM context, we have methods that are truly predictive, that we can say with confidence that a particular mechanism is or is not consistent with experiment. And that helps us to rule out the large number of incorrect mechanisms in the biochemical literature and identify the true mechanisms and the true transition states for enzyme catalyzed reactions. Charismate mutase is a poster child for us. It's a a nice guinea pig application that I think bears some repetition as a test for computational methods. And we can compare, we can test, for example, QM-MM calculations against full QM calculations. This has worked from a few years ago where we optimized the structure of the transition state in fully quantum calculations with 2000 atoms within our QM region. And the results agree quite well, very well between QM and QM-MM. So in general, for modeling enzyme reactions, it makes sense to focus your computational effort on the active site and treat the rest of the system more approximately in the QM-MM framework. We can also account for catalysis. So we can account for barrier lowering by the enzyme, not just the absolute barrier, but the difference in the reaction barrier for the reaction taking place in water and in enzyme. This is very simple for the case of charismatic mutase because this is a unimolecular reaction. It takes place in water. The rearrangement is slow but observable in aqueous solution. And we can do multiple QM-MM calculations of reaction barriers in water and in enzyme and find that the QM-MM calculations reproduce the barrier lowering that is observed experimentally by for the enzyme. So what makes the enzyme a good catalyst? Well, it goes back to the idea of Pauling of transition state stabilization. This blob here shows the difference in electrostatic potential between the ground state, the reactant, and the transition state for the enzyme catalyzed reaction. And where this blob is not green, where it is red or blue, there are significant differences in electrostatic property between the transition state and the ground state. And the enzyme places charged groups specifically to recognize these differences between the ground state and the transition state. It is literally encompassing and embodying the principles first put forward by Pauling of transition state stabilization. And of course, calculations allow us to study transition states within enzymes and see how they're stabilized. And our other prototype example, PHPH, work again with Walter Thiel, we were able to show that for this OH group in flight in this aromatic substitution reaction, a hydrogen bond is formed over here with a conserved proline residue. That's only found in the transition state of the reaction. So that specifically stabilizes the transition state and lowers the barrier to reaction. So those are examples of how we can understand not only the reaction mechanism, but catalysis through QM-MM calculations. But it is important to bear in mind that we have to be aware of the potential limitations of the QM and QM-MM methods that we're using. And the workhorse for many calculations in biological and non-biological context nowadays is density functional theory. So looking at QM-MM calculations with a range of density functionals over here, this is calculating the barrier for reaction for charismatic mutase, you can see that the barrier is significantly different with different functionals. They're all perfectly respectable functionals, V3 lip here in the middle, and we have PBE and we have B half and half lip. None of those functionals a priori can be preferred over any of the others. They're all equally theoretically sound or unsound, if you prefer. There is no rigorous way to improve those calculations, unlike an ab initio calculation, where we can reduce the degree of approximation to the Schrödinger equation, increase other aspects of the simulation. What we're able to do, so the previous calculations I showed you, the couple cluster calculations, employed a complex but usable scheme on localization to allow for the very computationally demanding couple cluster method. This can now be done in a much more straightforward way through so-called projector-based embedding. And this is a multi-scale calculation, if you like. This has an ab initio calculation within a DFT calculation within a QM-MM context. This removes the dependence of the energy barrier on the choice of density functional. So when we do a projector-based embedding ab initio calculation of the reaction barrier over here on the right, with an ab initio method embedding in DFT, the barrier is invariant depending on the functional. So now we can use this straightforward projector-based embedding calculation, calculate a DFT QMMM profile, and using the ab initio projector-based embedding, we can calculate the barrier with confidence and remove the uncertainty due to density functional theory. And this is, I think, usable and useful not only in a biological context, but also in non-biological applications. Wherever DFT is uncertain, this is a way to help correct energy profiles calculated with those methods. This is another enzyme example, but just to show this is applicable for a metalloenzyme. This is Rubisco, so it's a magnesium-containing system. And this is the activation energy, and this is the reaction energy over here with DFT, and you can see different DFT functionals give rather different results. But when we do embedding, when we embed an ab initio calculation within DFT, we can remove that sensitivity to the functional and predict the barrier and the reaction energy with confidence using high levels of QMMM theory. So QMMM calculations can be highly accurate. We have access now to the best QM methods that can be used for modeling the reactions of small molecules. And that's really the beauty of QMMM, that we can focus our computational effort on the active site, as I say. Low-level QMMM calculations have an important role to play. One is that it is possible to simulate the dynamics of enzymes using low-level QMMM calculations. So it's not a case where one always has to reach for the biggest gun in the armory. It's always a good idea to examine the dynamics of an enzyme. And in some cases, that's absolutely crucial to understand reactivity. So we've applied QMMM methods in a number of contexts where we've made predictions which have been validated and tested by experiment, which of course is the acid test of whether these calculations are useful. So this is another enzyme called phosphate dehydrogenase. We ran simulations of this enzyme. It's of interest for cofactor recycling, so it's potentially important for biotechnology. And what we found in our QMMM simulations is that this is the transition state, and only in the transition state, there was an interaction between this group down here, a methionine, a sulfur-containing amino acid, and the histidine, which becomes positively charged during the reaction. That interaction, which stabilizes the histidine, is only found in the transition state. It is a catalytic interaction, just like the interactions I mentioned previously, for PHBH and charismatic mutase. So the question is this something which is a real and important biochemical catalytic interaction? Before this work was done, methionine was not believed to have any direct catalytic role. And our collaborators, led by Wilfred van der Donk in the US, were able to do experiments in which they replaced the methionine by natural and unnatural amino acids. And they found that when they did so, the rate of reaction went down, but nothing else was affected. So it is indeed a truly catalytic interaction. And when we go back and look in the protein data bank, we find that this is an interaction which does occur in other systems. So it's potentially important. So that was a nice example of a prediction from a QMMM simulation of a reaction in an enzyme being borne out by experiment. We've also used QMMM methods to look at other effects in catalysis. So this is the example of aromatic amine dehydrogenase. This is work where Nigel Scrutton's group in Manchester did some beautiful structural and biochemical experiments. And we were able to use QMMM methods to calculate kinetic isotope effects for the primary kinetic isotope effect for transfer of this proton, which is the rate limiting step in the reaction. And we were able to find, using the apparatus developed by Don Trular, variational transition state theory calculations, we were able to calculate kinetic isotope effects in good agreement with experiment. And they're rather large, implying rather a large contribution from tunneling. What the calculations are able to do here is to dissect and analyze the catalytic contribution of tunneling. So we're able, to switch on or switch off tunneling in the computer. So we're able to say what is the contribution of quantum tunneling to reducing the reaction barrier. And it's significant, but not enormous. And it fits well, again, within a transition state theory context. And an application more in the drug discovery field. So there's great interest in finding targeted covalent inhibitors for proteins and enzymes. And one particular target is this enzyme fatty acid amide hydrolase. And what we were able to do here was we predicted which way round this inhibitor was likely to bind in this binding site of the enzyme. Effectively, this inhibitor can bind pointing up or pointing down. Both of those are perfectly stable and reasonable. In fact, they probably are both populated experimentally, but only one of them is able to react efficiently according to our QMMM calculations. So we predicted the binding mode, the covalent binding mode from reaction simulations of this enzyme, and they were borne out when the crystal structure was solved a couple of years later on. So this is again a nice example of a prediction from QMMM modeling borne out by experimental measurements. And we're using QMMM methods now to target other enzymes and other proteins. And this is one topical example. We're modeling with Vicente Moliner and Alessio Lodola. Vicente did some very nice calculations on the substrate and now also on the inhibitor, where we've looked at the reaction mechanism of a known inhibitor and then suggested alterations that may change its chemistry, synthetically accessible alterations that alter the degree of reversibility of the inhibitor. And those predictions are now being tested experimentally as a potential route to discovering inhibitors of that enzyme as potential antivirals. Another pertinent recent example is antibiotic resistance. So I think we should all be concerned about antibiotic resistance. It hasn't gone away in the pandemic. In fact, it has become worse because of the increased use of antibiotics. And antibiotic resistance, it's not an understatement to say it threatens many aspects of modern medicine, not least cancer therapy, transplant therapy. So this is not just about cuts and bruises. This is about how antibiotics may or may not be usable throughout the edifice of modern medicine. One particular threat is the resistance to carbapenem antibiotics. So I mentioned meropenem at the beginning, discovered by Zeneca, approved in 1994. And of course, when we use an antibiotic, resistance appears. And this is now a significant threat to the use of drugs like meropenem in the clinic. And if you're unlucky enough to get a resistant infection, E. coli, for example, then your chances of survival are, even in the best healthcare systems in the world, are only 2 in 3. Not very good. And these are significant threat and widening threat to medicine. So we want to use QMMM methods to understand, and we hope predict, antibiotic resistance, perhaps to help the development of new antibiotics and new inhibitors. Now, these enzymes, these antibiotics are broken down by enzymes called beta-lactamases. Beta-lactamases, the older beta-lactamase enzymes, which destroyed things like penicillin, were vulnerable to meropenem and the other carbapenems because they are inhibited. They don't turn over the antibiotic. Unfortunately, more recent enzymes, things like KPC, lots of acronyms, are able to break down carbapenems efficiently. They destroy the covalent complex, the acyl enzyme, with a low barrier. So they destroy the antibiotic quickly. So we wanted to know if we can use QMMM calculations as the equivalent, the computational equivalent of a biological assay. Can we predict whether enzymes can break down a particular antibiotic or not? So we modeled the reaction. We believe that we've identified the reaction mechanism going from the acyl enzyme to the tetrahedral intermediate in this breakdown of the covalent complex. And what we found was that we were able to distinguish by QMMM calculations between the new enzymes, those in red, which are able to break down carbapenems, and these in blue over here, which cannot, the older enzymes. So there's nothing parameterizing these calculations. This is a first principles calculation of the barrier in a QMMM molecular dynamics simulation. And we can distinguish between enzymes that do and do not break down a particular antibiotic. It took us about 20 days to do these simulations for each enzyme with 3 repeats of each, which is fine, but it's not quite fast enough to compete with an experimental assay. So what we did was to reduce the amount of calculation we did, either just following the minimum energy path and then reducing the amount of molecular dynamics simulations. And by doing that, we don't degrade the quality of the prediction, and we're able to make a QMMM prediction of the activity, the biological activity of these enzymes within about 4 hours per enzyme. And that is now competitive with experimental assays, and of course much cheaper. And it is a computational assay of drug resistance. But more than that, these calculations, because they reproduce the experimental activity correctly, they're telling us at some level why some enzymes allow antibiotic resistance and others don't. The molecular details of catalysis reveal the difference in antibiotic resistance. So an example of this, so this is work by Vivi Hirvonen again. What she did was to look at 2 different class D beta-lactamases. It's a slightly different type of beta-lactamase, and look at the activity of these enzymes against a different class of antibiotic, in this case, cephalosporins. These 2 nasty enzymes differ in their ability to break down a cephalosporin antibiotic. Vivi was able to use QMMM simulations to calculate the barrier to reaction, which correctly reproduce the difference in activity that's observed in the lab and the clinic in the resistance profile for these 2 enzymes, and able beyond that to show that the difference in activity comes from very subtle changes in the active site, a small change in hydration of the catalytic base, small change in the number of water molecules present, which significantly affects the activity of the enzyme. Those subtle differences we can target potentially in inhibitor design or in antibiotic design to combat antibiotic resistance. So a slight change of pace. So it would be wonderful if everything boiled down to the active site of an enzyme, if we could just focus on the small part of the enzyme where the reaction happens. But enzymes are big, and they're big for a reason. If you look at the catalytic proficiency of enzymes against their molecular weight, there's a reasonably good correlation. Enzymes that do difficult chemistry on the whole are bigger. Now, there are a couple of reasons for that. One is the folding energy that's required to organize the active site. But an emerging frontier is that this also relates to the heat capacity of the enzyme and the change in heat capacity that's associated with reaction. We believe from emerging experimental and simulation efforts that this activation heat capacity is truly fundamental, and it explains something rather odd about enzymes. So enzymes are observed in many cases to show temperature dependence, which is definitely not Arrhenius. It's not linear. You increase the temperature, and there is a point at which temperature at which the activity is maximal, and then it drops off beyond that. And this is not in general due to protein unfolding, which is the explanation in many textbooks. This is most clear for enzymes adapted to low temperatures, which lose activity at higher temperatures but remain very well folded. This can be explained by what we call macromolecular rate theory. If we invoke a negative activation heat capacity, this explains why and how these rate profiles are curved, there's a drop in the flexibility of the enzyme complex between the reactant complex and the transition state complex. And we're able to reveal this by simulations. So this is one enzyme called ketosteroidisomerase. And what we see is a clear difference in the dynamics between the reactant complex and the transition state complex. But this is not a simple tightening across the whole enzyme in some homogeneous fashion. It is in different regions of the enzyme, and some regions become more mobile and some less mobile in the transition state complex. We're able to use simulations from the definition of heat capacity. We can calculate the fluctuation in energy during these MD simulations of transition states and reactant states, and derive the heat capacities and find heat capacities in very good agreement with experiment. So potentially these simulations can guide engineering of these enzymes to change their optimum temperatures, even at the level of a single hydrogen bond changing the optimum temperature. And this goes some way also to explain why enzymes are large. We find significant contributions from distant parts of the protein, not just limited to the active site. This is also apparent when we look at the evolution of a designed protein catalyst. This is a Kemp eliminase, and a Kemp eliminase, so the reason for choosing Kemp elimination is this is a reaction that doesn't occur in nature, and effective protein catalysts can be designed to do it. This is an example, and this is the rate profile of the designed catalyst. As Francis Arnold and Don Hilvert and others have shown, directed evolution is a very powerful technique to improve catalysts, and this is indeed the case here. So work done by Adrian Bunzel in Don Hilvert's group indeed showed that it was possible to improve the catalytic activity of the enzyme quite significantly. What was unexpected was that that led to a curved temperature dependence, the emergence of a negative activation heat capacity. And we were able to do simulations that reproduce this, that reproduce the change in heat capacity between the design and the evolved variant. And these are MD simulations, again, of the ground state and the transition state. And if you squint, you can see that the evolved variant on the right closes more. This is the active site, and these loops are closing over the active site. So that looks more like a natural enzyme. And we're able to analyze these changes in behavior. And really this arises from more correlation. So what evolution has done is to introduce correlations in the transition state ensemble that really significantly tighten up the ensemble related to its increased catalytic power. This is the design, this is the design over here. This is the evolved variant. You can see there are many more lines, many more connections. And what's interesting is that the networks that we find predict positions that are found by evolution. So they predict distal mutations that arise in further improvement of the enzyme's catalytic activity. So these simulations are potentially a guide to enzyme engineering and design. So I'll finish off by talking about how I think the way we interact with our simulations is also changing, and changing quite radically. So there's been a lot of hype about virtual reality for many years, but it is now a technology which is feasible and usable by the practicing chemist or biochemist. And this is a way potentially for us to collaborate, even in lockdown, for example, to collaborate together on molecular models, and not just molecular models, but molecular dynamic simulations. So this is my student, Becca Walters, who's done very nice work with Helen Deeks on showing that we can use virtual reality interacting with the molecular dynamic simulation. You can see that this protein is waggling around, and we've again used this to look at binding of inhibitors to the SARS-CoV-2 main protease. And we're also using it to look at other catalysts. This is a zeolite work done by Simon Benny, Joe Crossley-Lewis, looking at how promoters bind within a zeolite framework. And this is a an approach, the virtual reality molecular dynamics approach, which is very attractive and useful for education. So just to give you an example, I hope that this video will play. This is a class that we run for undergraduates at Bristol now, using interactive MD in VR. This is charismatic mutase again. And this is a chemical reaction being driven in virtual reality. The student here is forming the chemical bond, making the glycine rearrangement happen in VR using an approximate density functional theory calculation. And students with no training are also able to take the protein, remove the reactant, and to bind it back into the active site to find the correct location and the correct conformation of that molecule within the protein. I think something here to notice is the dynamic response of the protein, the way that those charged groups swing around to interact with the protein, to interact with the reactant, and potentially then to stabilize the reaction as it takes place. So I think that's the future of how we will interact with our simulations. I'll stop these before they take over my computer even more. And just to finish with, these, as I say, these are methods which are intuitive and accessible. Some of you experienced interactive molecular dynamics in virtual reality. And this is JMT. JMT was very interested when we took him in VR, and here he is manipulating a molecular structure of, I think, a zeolite catalyst in this case. And really it shows that it's possible for anyone to use this sort of technology to start to explore chemistry and catalysis in molecular and dynamic detail, really very intuitively and accessibly. So I'd like to finish off by thanking the people who've funded me, and particularly to thank the people who've done the work, and also to thank you for your attention. Thank you very much.
Good. Well, thanks very much, Adrian. I'm sorry, I can't turn my video camera on now. An absolutely wonderful lecture, really showing the power of computational methods in this field. So we do have a few moments for questions. So please, everyone, questions, either put your virtual hand up or put something in the Q&A. I mean, I'll kick off, Adrian. I mean, what has been achieved with QMMM is hugely impressive. What do you think are the next challenges in the development of QMMM techniques?
I think that's a very good question. And of course, as you well know, certainly not limited to enzymes. And the principles, I think, are, or the problems are the same for non-biological systems too. I think it is the coupling of sampling with the modeling of reactivity to be able to use high-level methods to get accurate chemistry. We're never going to be able to do molecular dynamic simulations with a couple cluster method. Instead, we have to use some sort of multi-scale framework to sample at different levels. I think those tools are now becoming available. And I think the challenge is for us to understand, in the case of enzymes, why it is that we see conformational changes, why it is that we see dynamical changes, why has evolution chosen to make these changes? And I think that provides a guide for us, not only in engineering enzymes, but in designing and engineering non-biological catalysts, heterogeneous and homogeneous in future.
Thanks very much, Adrian. Are there other questions?
So Mike has a question.
Mike, please go ahead.
Can you hear me?
Yes,
Yeah.
Mike.
Hello, Adrian. Let me reinforce what Richard said. A wonderful talk, a beautiful talk. I really enjoyed that. But there's a but coming. as you might expect. The but is, obviously you use different levels of calculation as you extend away from the active site in this proteinaceous entity. And you said how important that outer structure was towards the end of your talk. Whereas at least in the earlier level of modeling, I think you implied that you use a lower level of calculation to determine that interaction. Is there an ambiguity there, or is there not?
That's a very good question. I would say that we have tested. When we're looking, for example, at the Kemp eliminase, we have looked at QMMM calculations. I didn't show this, of the reaction barrier in the design and in the evolved variant, and they capture the difference in barrier. So that implies that for whatever reason, the QMMM approximation is working well. I think that is because the effects of long-range electrostatics are captured reasonably well, even by an atomic point charge model of the protein environment. And the crucial thing is capturing the change in structure. And this goes back to Richard's question of the challenge. Well, the challenge is to find these complexes which dominate reactivity. So the structure of the enzyme varies, and it appears that there are certain configurations which are much more reactive than others, and those clearly will dominate reactivity. They're accessible, they're thermally accessible configurations. So it's finding those catalytic conformations and analyzing their properties. While we can't yet do MD simulations with a full quantum method, we know that those barriers are similar if we calculate with a full quantum method and a QMMM method. But it's that initial sampling, the sampling of the initial conditions and finding those reactive conformations, which I think is crucial. And I don't see a practical way to do that at higher levels at the moment.
Thank you. Thank you.
Thanks. There's a question from Glenn Sunley, who first congratulates you on the JMT medal and the great talk. He says, we've discussed a number of times whether it's a heat capacity that changes with temperature or is it an entropy issue? I think the two are related, aren't they? I think in zeolite catalysis, we would pin it on entropy. Any comments?
That's a fundamentally important question, and we're working with Glenn on these promoter systems, PP, and one of the things that Glenn and I are both interested in is understanding if we can use principles from biological catalysis, such as perhaps the optimization of entropy in design and development of non-biological systems. There is certainly an entropy effect. That's part of enzyme thermoadaptation. There's an entropy trade-off. What macromolecular rate theory does is, of course, to go beyond that, because entropy actually in these complex systems is not temperature independent. It changes with temperature. So it stops being a good guide to what evolution has optimized and to what's driving catalysis. So in some cases, the contributions of enthalpy and entropy for an enzyme may be significantly different at different temperatures beyond just the T delta S effects. But it's certainly true that evolution does change activation entropy in many cases.
Well, thanks for that. And I think the comment that both you and Glenn have made as to whether these effects are important in zeolites is a particularly interesting one. I think we can take, if there is one more question, we can take one. But if not, could it just remains for me to thank Adrian again for an excellent lecture, a really stimulating talk, and once again, congratulate you, Adrian, on the award of the 2020 John Marriott Thomas Lecture. So congratulations, Adrian. Thanks very much. So that ends the session.